per our early discussion, we have discussed that the plasma membrane surrounds the cell and it creates a selectively permeability. Now, in this slide, we will discuss about the function of plasma membrane. Plasma membrane forms a specialized compartment by selectively permeability, and that selectively permeability is responsible for unique environment, creation of concentration gradient from outside to inside, creating differences in pH and the negative charge. That is also responsible for asymmetric protein distribution, cell-cell recognition, site for the receptor molecules, binding, which is responsible for the cell signaling of drug, hormones and other chemicals, that is responsible for intracellular reactions. Cell membrane is also responsible for the controlling and regulating the reaction sequences which is generated through enzymes on a particular substrate, and which is responsible for the line-upping of enzyme in the proper sequence. Along with all, the major function of plasma membrane is transportation of molecule from inside to outside and outside to inside. Cell collects its required molecules and ions from surrounding extracellular fluids or ECF. There is an unceasing traffic of molecules and ions in and out of the cell through its plasma membrane. Some molecules are glucose, sodium, calcium. In eukaryotic cells, there is also transportation in and out of membrane-mounted intracellular compartments such as nucleus, endoplasmic reticulum, and mitochondria. And the transported molecules are protein, mRNA, calcium ion, ATP. In transportation of cell membrane, there are two problems to be considered. First is relative concentration. The molecules and ions move spontaneously down their concentration gradients. That is, from a region of the higher concentration to the lower concentration, and the process is known as diffusion. Molecules and ions can be moved against their concentration gradient. But in this process, which is known as active transportation or active transport, which require the expenditure of ATP to get the energy. Second consideration is lipid bilayers are impermeable to most of the essential molecules, ions and drugs. The lipid bilayer is permeable to water molecules and a few other small uncharged molecules like oxygen, carbon dioxide. These diffuse freely in and out of the cell. The diffusion of water through plasma membrane is of such importance and to the cell that is given a special name, which is known as osmosis. Lipid bilayers are not permeable to ions such as cations like potassium, sodium and calcium, and anions like chloride, bicarbonate. It is also impermeable to small hydrophilic molecules like glucose and large molecules or macromolecules like protein and diffusion, also called as non-ionic transport or passive diffusion. The driving force for this process is the concentration or electrochemical gradient. The difference of concentration of the molecules on either sides of the membrane. Movement is a result of kinetic energy of the molecule. Since no energy source is required, the process is called passive diffusion. During this passive diffusion, the molecules present in the aqueous solution at the absorption site partition and dissolve in the lipid material present in the membrane, and finally leaves it by dissolving again in the aqueous medium of cytosol inside the membrane. The passive diffusion is expressed by fixed first law of diffusion, which states that molecules diffuses from a region of higher concentration to the lower concentration until the equilibrium is attained, and the rate of diffusion is directly proportional to the concentration gradient across the membrane. The passive diffusion process is a downhill process, that is, moves along the concentration gradient. The process is energy independent. The rate of transfer is directly proportional to the concentration gradient. Greater the area of cell membrane and lesser the thickness of the membrane, faster the rate of diffusion. That is why in animals, thus more rapid is the rate of absorption from the intestine than the stomach. The rate of transport of unionized species is 3 to 4 times more than the ionized species. The membrane is lipidal in nature, so the greater the membrane water partition coefficient of the molecules, faster the rate of absorption. Now let's see the factors affecting diffusion across the plasma membrane. Diffusion is actually regulated by a number of factors. The greater the lipid solubility of the molecule, the more permeable to the plasma membrane. Oxygen, water, carbon dioxide rapidly diffuses across the lipid bilayer. Plasma membrane is a semi-permeable, but water, while polar in nature, is small enough to freely move across the plasma membrane. Larger hydrophilic uncharged molecules, such as sugars, do not freely diffuse. Charged molecule cannot diffuse through the lipid bilayer. They have different channels. Ion channels and specific transporters are required for the charged molecules and larger uncharged molecules. Osmosis is a special term used for the diffusion of water through the cell membranes. Although water is a polar molecule, it is able to pass through the lipid bilayer of plasma membrane as because of aquaporins, transmembrane proteins that form hydrophilic channel greatly accelerate the process, but even without this, water is still able to get through. Water passes by diffusion from the region of higher to a region of lower concentration. Please note that this refers to the concentration of the water, not the concentration of any solute present in the water. Water is never transported actively. That is, it never moves against its concentration gradient. However, the concentration of water can be spurred by active transport of solutes. In this way, the movement of water in and out of the cell can be controlled. For example, the reabsorption of water from the kidney, the tubules back into the blood depends on the water following behind the active transport of sodium. Now let's discuss about the solution types relative to the cell. In this regard, solutions are of three types: hypertonic solution, hypotonic solution, and isotonic solution. In case of the hypertonic solution, the concentration of the solution is higher than the cell. More dissolved particles outside of the cell than the inside of the cell. Hyper means more and tonic means the dissolved particles. Water moves out of the cell into the solution and ultimately cell shrinks. If a red blood cell are placed in the sea water, which contains about 3% of the salt, they lose water by osmosis and the cells shrivel up. Sea water is hypertonic to their cytoplasm. Similarly, if a plant tissue is placed in the sea water, the cell contents shrink away from the rigid cell wall. This process is called plasmolysis. If we place a cell without cell wall, a plant cell without cell wall in 13% mannitol solution, then the thing is known as plasmolyticum or osmolyticum or osmotic stabilizer, which can be introduced in case of the protoplast fusion technique. Seawater is also hypertonic to the extracellular fluid of the most marine vertebrate. To avoid fatal dehydration, these animals must continuously drink seawater and then desalt it by pumping out of their gills by active transport. Marine birds, which may pass long periods of time away from the fresh water, and the sea turtles uses a similar device. They too drink salt water to take care of their water need and use metabolic energy to desalt it. In the herring gull, the salt is excreted by two glands which are located in the head and released to the outside through the nostrils. Marine snakes use a similar desalting mechanism. Now come to the hypotonic solution. Hypo means less, tonic means dissolved particles. Solute concentration of solution lower than the cell, less dissolved particles outside of the cell than the inside of the cell. Water moves into the cell from the outside solution and ultimately cell expands and may burst. If the concentration of the water in the medium surrounding of the cell is greater, for example, the red blood cell when placed in a hypotonic solution, for example in pure water, it bursts immediately from the influx of water. Plant cells and the bacterial cells avoid bursting in the hypotonic solution surrounding by their strong cell walls. These allow the buildup of targar within the cell. When the targar pressure equals to the osmotic pressure, osmosis ceases. In case of the isotonic solution, the solute concentration of solution equal to that of the cell. No net water movement. When red blood cells are placed in 0.9% salt solution, they neither gain or nor lose water by osmosis. Such a solution is said to be isotonic. The extracellular fluid of the mammalian cell is isotonic to their cytoplasm. This balance must be actively maintained because of large number of organic molecules dissolved in the cytosol, but not present in the ECA. These organic molecules exert an osmotic effect that, if not compensated for, would cause the cell to take in so much of water that it would swell and might even burst. This fate is avoided by pumping sodium ions out of the cell with the sodium potassium ATPase pump. The osmosis is very important for human health. Large volume of the solution of 5% human albumin are injected into the people undergoing a procedure called plasmapheresis. The albumin is dissolved in physiological saline, that is 0.9% NaCl, and is therefore isotonic to the human plasma. The large protein molecules of the albumin have only a small osmotic effect. If 5% solutions are unavailable, pharmacists may substitute a proper dilution of a 25% albumin solution, mixing one part of the 25% solution with 4 parts of diluent result in the correct of the 5% solution of albumin. But in several cases, the diluent used was sterile water, not a physiological saline, so the resulting solution was strongly hypotonic to the human plasma. The result is massive life-threatening hemolysis of the patient. Osmosis produces a physical force which helps in the movement of water into a cell, can put pressure on the plasma membrane. Animal cell will expand and may burst. Organisms with cell wall, such as plant, do not burst, as because cell membrane pushes against the cell wall. The rigid wall resists due to its own structural integrity. These opposing forces create turgidity, which keeps the plant upright. If you don't water a plant, it wilts. This is called plasmolysis. Water it and the leaves will come back up to the re-establishment of the turgidity. Here you can see the fate of the plant cell and different concentration of the salt solution, and about the animal cell like RBC in different concentration of the salt solution. Another transport mechanism is facilitated diffusion, which allows the diffusion of the large membrane insoluble compounds such as sugars and amino acids. The process doesn't require energy. For this reason we call it passive. It is a carrier mediated downhill transport system, but much faster than the passive diffusion. Since no energy is required, the process is not inhibited by metabolic poison that interfere with the energy production. Transport of the glucose into RBC is the example of the facilitated diffusion. Classic example of the facilitated diffusion is the GI absorption of vitamin B complexes, especially vitamin B12, an intrinsic protein or intrinsic factor, a glycoprotein produced by the gastric parietal cell, forms a complex with vitamin B12, which is then transported across the membrane by carrier system. Movement rate of particle with saturate, maximum rate limited by number of transporters. Once all transporters are operating at 100%, an increase in the concentration will not increase the rate. Here you can see a carrier mediated downhill transport system. The blue color receptor is a carrier. In course of the transportation of the glucose binds to the transport protein. Transporter changes conformations and glucose is released into the cell. The intracellular glucose is immediately phosphorylated. When that becomes phosphorylated, the phosphorylated glucose doesn't diffuse out because their structure is quite different and larger. And remember that transport protein is very specific to the structure of glucose, not the phosphorylated one. Internal glucose which are unphosphorylated, the concentration remain low, providing a large concentration difference for the entry of the glucose from the outside to the inside. Another important transport mechanism is active transport. This transport mechanism requires energy in the form of ATP. The transport of molecules are against the concentration gradient, so the process is known as uphill process. It requires energy, and active transport proteins are highly selective. Actually, there are a number of different active transport systems are available. First one is the primary active transport. In this process, there is a direct ATP requirement. The process transfers only one ion or molecules in one direction, hence called uniport system. The carrier proteins involved in the primary active transports are of two types. One is the ion transporter, which are responsible for the transporting ions in or out of the cells. A classic example of ATP-driven ion pump is proton pump, which is implicated in the acidification of intracellular components. Two types of ion transporters are available: organic anion transporter and organic cation transporter. Another type of transporter is ABC or ATP binding cassette transporter, which is responsible for transporting small foreign molecules like drugs and toxins, especially out of the cells. So we call it exhaustion or efflux pump, which make them clinically significant. PGP is an example of ABC transporter. PGP or P-glycoprotein is responsible for the pumping hydrophobic drug, especially anti-cancer drug, out of the cell. The presence of the large quantity of PGP makes cell resistance to the several drugs used in chemotherapy. ABC transporter present in the brain is responsible for the pumping wide range of the drug out of the brain. Second active transportation system is secondary active transport. In this process, there is no direct requirement of the ATP. That is, it takes advantages of previously existing concentration gradient. The energy required in this transporting an ion aids transport of another ion or molecule, either in the same direction or in the opposite direction. This is divided into two categories: symport or co-transport. It involves the movement of both the molecule in the same direction. Sodium and glucose symporter uses the potential energy of the sodium concentration gradient to move the glucose against its concentration gradient. Another thing is peptide transporter or H+ coupled peptide transporter, which is implicated in the intestinal absorption of the peptide like drugs like beta-lactam antibiotics. Next method of the secondary active transport is antipode or counter transport. It involves the movement of the molecules in the opposite direction. For example, expulsion of the hydrogen ion using sodium ion gradient in kidney. Active transport is the faster than the passive diffusion. The process requires ATP burning. So ATP plays the role of the pumping. It can be inhibited by metabolic poison that interfere with the energy production like fluoride, cyanide, dinitrophenol or lack of oxygen. Active transport system is important in the renal and the biliary excretion of the many drugs and their metabolites. In short, we can explain this process as a phosphate group is transferred from the ATP to the transporter protein and the protein energized by the added phosphate group. The energized transport protein now changes its conformational change and its shape, and it releases the molecule that is to be transferred. We can denote it by X on the other side of the cell. The phosphate group is now released from the transport protein and it resumes to its original shape and the process repeats. Another method of transport of the large particles is bulk flow. Vesicles are used to transport large particles across the plasma membrane. In this image, you can see that the internal vesicle containing particles fuse to the cell membrane and ultimately release the content outside to the cell. Bulk flow may be of two types, exocytosis and endocytosis. In exocytosis, the cytoplasmic vesicle merges with the plasma membrane and releases its content. For example, Golgi body vesicle merges with the plasma membrane and releases their content. In endocytosis, it involves engulfing of extracellular material within a segment of the cell membrane to form a vesicle, which is then pinched off intracellularly. This phenomenon is responsible for the cellular uptake of macromolecular nutrients like fat, starch, oil, soluble vitamins like A, D, E, K. The process is divided into two types. One is the phagocytosis or cell eating, which is related to the absorptive uptake of the solid particles. And another is the pinocytosis or cell drinking, which uptakes of solutes. Orally administered polio vaccine, large protein molecules and the botulism toxin are thought to be absorbed by the pinocytosis process. Sometimes an endocytic vesicle is transferred from one extracellular compartment to another. Such phenomenon is called as transcytosis. Another process of endocytosis is the receptor-mediated endocytosis. Receptor proteins on the plasma membrane bind specific substances like vitamins or hormones. Then the membrane sinks in and forms a pit called coated pit. Pit pinches flows to form a vesicle around bound substances. Cytoskeleton aids in pulling the membrane up and the vesicle formation. In next slide, we may see the image of the carrier-mediated endocytosis or receptor-mediated endocytosis. In this image, on the left side, you can see the animated image of receptor mediated endocytosis, where the red color white shaped structures denotes the receptor. In the right side, you can see the electron micrograph of this process.